So this is the classical complement pathway, which is antigen dependent or antibody dependent. So it needs 2 molecules of IgG or 1 molecule of IgM. So the way that this works is you have what's called the QR2S2, which binds 1 molecule of IgM or 2 molecules of IgG. So I have the 2 molecules of IgG right here. This causes the release of the C1 inhibitor, which then activates C1R. C1R cleaves and activates C1S, and then C1S cleaves C4 into C4A and C4B. So I'll draw that out here. Let's make this a little bigger. So we got this C4B right here, okay? So that is our C4B, or C4 I mean, which will come in and get cleaved, and we're going to put them all the way up here. So now we're interested in C4B. Okay, so C2 now will complex with C4B, which is cleaved by C1S. So C2 will now join in the party and complex with this. So now you have C4B, C2. So this guy is now C4B, C2. After that, you have cleavage of that by C1S, and then that gives you C, so C1S comes in here, C1S comes in, cleaves this, right? And then you have what is called C4B2A, which is also known as C3 convertase. This is a little bit different than the C3 convertase that we saw in the alternative pathway, right? So after that, this acts as a C3 convertase, so it cleaves C3 into C3A and C3B. This can now complex with C3B, so C3B, after it's been cleaved by C3 convertase, will come in and join. That will yield C5 convertase. Now remember what C5 convertase does is it takes C5 and turns it into C5A and C5B. We're interested in C5B here because it complexes with C6, which recruits C7, C8, and C9. The actual molecule of C5B678 will promote C9 polymerization, which forms the pore in the MAC complex, the MAC attack complex. So if we have this MAC attack complex here, right, this is our pore, and then we have a pore formed around here, like this, by the C9 complex. And then you have, if you have your bacteria in here, this is your bacteria cell wall, all the contents are going to start leaking out. So that is the classical activation pathway. In the next video, I'm going to talk about why this doesn't happen in all of our human cells. Why doesn't complement just bind to all of our human cells and start killing them? So I'm going to talk about the regulation of complement pathways.